Hello and welcome back. We're going to look at the electron transport chain in a bit more detail because I felt like it needed a presentation of its own. So here we have the mitochondria. And where does the electron transport chain take place? Well, it actually takes place along these membranes here called cristae. And here we have the intermembrane space, which is between the cristae and the outer membrane of the mitochondria. So what actually happens? Well, we mentioned in the Krebs cycle that reduced NAD is made and it will be useful in this stage, the electron transport chain. So what happens with NADH is here it's actually oxidized back to NAD. It releases a hydrogen atom, also known as proton, and this proton moves through a proton pump. And it requires energy to do this because it's moving the protons from low concentration to high concentration. Where does it get this energy from? Well, it gets the energy from the electrons that were also made in the Krebs cycle previously. These electrons bounce from this electron carrier 1 to electron carrier 2 to electron carrier 3 until they get to oxygen, which is the final electron acceptor. The oxygen also combines with two protons here, two hydrogens, and that combines to make water, which is one of our waste products of respiration. So the water that you breathe out as water vapour is made here. So if we go back to these protons or hydrogen molecules, atoms that are pumped through into the intermembrane space, they build up here. They build up their concentration level here, and they passively move from higher to lower concentration through this enzyme here, the ATP synthase. As they do this, this provides energy for ADP to combine with phosphate to make our ATP molecules in the numbers that were mentioned previously. So each glucose molecule makes around 38 molecules of ATP. Those ATP molecules that are made here in this electron transport chain are made by what is known as oxidative phosphorylation. So the phosphates are put onto ATP in the presence of oxygen here, and the ATPs that were made directly before in either glycolysis or the link reaction, well, Krebs cycle, those are basically called substrate level phosphorylations, and they are the two ways to make ATP.